Are you using retrograde tracers to map neural connections and still getting results that are more difficult to interpret than they should be? Even with a well-established tracer, interpretation can get complicated if the workflow around the tracer isn't well controlled. Here are 6 common problems that can make tracing results harder to interpret and the workflow variables to check before assuming the tracer is the issue. Number 1. Signal appears outside the intended target area. Retrograde tracers depend on controlled uptake at the injection site. In compact anatomical regions, small changes in injection placement, volume, or delivery technique can affect where the tracer is taken up. If the tracer spreads beyond the intended target, labeled neurons may reflect uptake from adjacent tissue, neighboring structures, or exposed fibers rather than the pathway being studied. That's why injection site verification and expected anatomy matter so much. Number 2. Labeling is weak, uneven, or hard to distinguish. Fluorescence is only useful if the labeled cells can be clearly separated from the surrounding background. Uneven labeling can complicate comparisons between animals, regions, or treatment groups. Before assuming that the tracer is responsible, evaluate tracer preparation, transport time, tissue condition, fixation and sectioning, imaging settings, and available controls. Weak or uneven signals can be especially problematic when the study depends on neuron counting, projection density comparisons, or clear identification of labeled cell bodies. Number 3. Background fluorescence makes the signal harder to trust. Background fluorescence can reduce confidence in retrograde tracing, especially when researchers need to identify individual labeled cell bodies or quantify labeled populations. Autofluorescence, excess tracer, nonspecific signal, section thickness, mounting conditions, exposure settings, and channel overlap can all reduce contrast. The goal is to improve the separation between true labeled neurons and background signal, because that separation determines whether the result can be interpreted or not. Number 4. Co-labeling creates channel or interpretation conflicts. Many retrograde tracing studies also use immunofluorescence, counterstains, reporter systems, or additional tracers. When a workflow includes multiple fluorescent signals, there are more opportunities for channel overlap and interpretation problems. To help avoid that, check filter sets, excitation and emission channels, spectral separation, tissue autofluorescence, and single label controls before imaging. Number 5. Labeled neurons are difficult to assign to a pathway. Sometimes cells are labeled, but the pathway interpretation is still unclear. This can happen when injection sites are near multiple projection targets, when anatomical boundaries are difficult to define, or when labeled neurons appear in unexpected regions. Tracer localization, anatomical controls, expected projection patterns, and the biological model all play a role in whether labeled neurons can be assigned to the intended pathway with sufficient anatomical support. Number 6. Results vary between animals or experimental runs. Retrograde tracing is sensitive to procedural details. Injection coordinates, volumes, delivery rates, tissue processing timelines, fixation and sectioning conditions, imaging settings, and analysis thresholds can all shift whether labeling appears consistent. When results are inconsistent, workflow documentation becomes part of the interpretation. The main takeaway is that retrograde tracing is not just about whether labeling appears. Clear interpretation depends on where the tracer was taken up, how the tissue was processed, and how the signal is imaged and analyzed at the endpoint. The article linked in the description walks through these 6 common retrograde tracing issues and the workflow variables behind them in more detail.